Variation and adaptation. Natural selection can only take place if there is variation or differences among individuals in a population. Importantly, these differences must have some genetic basis, otherwise selection would not lead to change in the next generation. This is critical because variation among individuals can be caused by non-genetic reasons, such as an individual being taller because of better nutrition rather than different genes. Genetic diversity in a population comes from two main sources: mutation and sexual reproduction. Mutation, a change in DNA, is the ultimate source of new alleles or new genetic variation in any population. An individual that has a mutated gene might have a different trait than other individuals in the population. However, this is not always the case. A mutation can have one of three outcomes on the organism's appearance or phenotype. A mutation may affect the phenotype of the organism in a way that gives it reduced fitness, lower likelihood of survival, resulting in fewer offspring. A mutation may produce a phenotype with a beneficial effect on fitness, and many mutations, called neutral mutations, will have no effect on fitness. Mutations may also have a whole range of effect sizes on the fitness of the organism that expresses them in their phenotype, from a small effect to a great effect. Sexual reproduction and crossing over in meiosis also lead to genetic diversity. When two parents reproduce, unique combinations of alleles assemble to produce unique genotypes and thus phenotypes in each of the offspring. A heritable trait that aids the survival and reproduction of an organism in its present environment is called an adaptation. An adaptation is a match of the organism to the environment. Adaptation to an environment comes about when a change in the range of genetic variation occurs over time that increases or maintains the match of the population with its environment. The variations in finch beaks shifted from generation to generation, providing adaptation to food availability. Whether or not a trait is favorable depends on the environment at the time. The same traits do not always have the same relative benefit or disadvantage because environmental conditions can change. For example, finches with large bills were benefited in one climate, while small bills were a disadvantage. In a different climate, the relationship reversed. Welcome back to Concepts of Biology. We'll continue to read Section 11.1, Patterns of Evolution. The evolution of species has resulted in enormous variation in form and function. When two species evolve in different directions from a common point, it is called divergent evolution. Such divergent evolution can be seen in the forms of the reproductive organs of flowering plants, which share the same basic anatomies. However, they can look very different as a result of selection in different physical environments and adaptation to different kinds of pollinators. Figure 11.5. Flowering plants evolved from a common ancestor. Notice that the dense blazing star and purple cone flower vary in appearance, yet both share a similar basic morphology. In other cases, similar phenotypes evolve independently in distantly related species. For example, flight has evolved in both bats and insects, and they both have structures we refer to as wings, which are adaptations to flight. The wings of bats and insects, however, evolved from very different original structures. When similar structures arise through evolution independently in different species, it is called convergent evolution. The wings of bats and insects are called analogous structures. They are similar in function and appearance, but do not share an origin in a common ancestor. Instead, they evolved independently in the two lineages. The wings of a hummingbird and an ostrich are homologous structures, meaning they share similarities despite their differences resulting from evolutionary divergence. The wings of hummingbirds and ostriches did not evolve independently in the hummingbird lineage and the ostrich lineage. They descended from a common ancestor with wings. The modern synthesis. The mechanisms of inheritance, genetics, were not understood at the time Darwin and Wallace were developing their idea of natural selection. This lack of understanding was a stumbling block to comprehending many aspects of evolution. In fact, blending inheritance was the predominant and incorrect genetic theory of the time, which made it difficult to understand how natural selection might operate. Darwin and Wallace were unaware of the genetics work by Austrian monk Gregor Mendel, which was published in 1866, not long after the publication of On the Origin of Species. Mendel's work was rediscovered in the early 20th century, at which time geneticists were rapidly coming to an understanding of the basics of inheritance. Initially, the newly discovered particulate nature of genes made it difficult for biologists to understand how gradual evolution could occur. But over the next few decades, genetics and evolution were integrated in what became known as the modern synthesis, the coherent understanding of the relationship between natural selection and genetics that took shape by the 1940s and is generally accepted today. In sum, the modern synthesis describes how evolutionary pressures, such as natural selection, can affect a population's genetic makeup and, in turn, how this can result in the gradual evolution of populations and species. The theory also connects this gradual change of a population over time, called microevolution, with the processes that gave rise to new species and higher taxonomic groups with widely divergent characters called macroevolution. Population genetics. Recall that a gene for a particular character may have several variants or alleles that code for different traits associated with that character. For example, in the ABO blood type system in humans, 3 alleles determine the particular blood type protein on the surface of the red blood cells. Each individual in a population of diploid organisms can only carry 2 alleles for a particular gene, but more than 2 may be present in the individuals that make up the population. Mendel followed alleles as they were inherited from parent to offspring. In the early 20th century, biologists began to study what happens to all the alleles in a population in a field of study known as population genetics. Until now, we have defined evolution as a change in the characteristics of a population of organisms, but behind that phenotypic change is genetic change. In population genetic terms, evolution is defined as a change in the frequency of an allele in a population. Using the ABO system as an example, the frequency of one of the alleles, I to the power A, is the number of copies of that allele divided by all the copies of the ABO gene in the population. For example, a study in Jordan found a frequency of allele A to be 26.1%. The B and O alleles made up 13.4% and 60.5% of the alleles respectively, and all of the frequencies add up to 100%. A change in this frequency over time would constitute evolution in the population. There are several ways the allele frequencies of a population can change. One of those ways is natural selection. If a given allele confers a phenotype that allows an individual to have more offspring that survive and reproduce, that allele, by virtue of being inherited by those offspring, will be in greater frequency in the next generation. Since allele frequencies always add up to 100%, an increase in the frequency of one allele always means a corresponding decrease in one or more of the other alleles. Highly beneficial alleles may, over a very few generations, become fixed in this way, meaning that every individual of the population will carry the allele. Similarly, detrimental alleles may be swiftly eliminated from the gene pool, the sum of all the alleles in a population. Part of the study of population genetics is tracking how selective forces change the allele frequencies in a population over time, which can give scientists clues regarding the selective forces that may be operating on a given population. The studies of changes in wing coloration in the peppered moth from mottled white to dark in response to soot-covered tree trunks and then back to mottled white when factories stopped producing so much soot is a classic example of studying evolution in natural populations. Figure 11.6. As the Industrial Revolution caused trees to darken from soot, darker colored peppered moths were better camouflaged than the lighter colored ones, which caused there to be more of the darker colored moths in the population. In the early 20th century, English mathematician Godfrey Hardy and German physician Wilhelm Weinberg independently provided an explanation for a somewhat counterintuitive concept. Hardy's original explanation was in response to a misunderstanding as to why a dominant allele, one that masks a recessive allele, should not increase in frequency in a population until it eliminated all the other alleles. The question resulted from a common confusion about what dominant means, but it forced Hardy, who was not even a biologist, to point out that if there are no factors that affect an allele frequency, those frequencies will remain constant from one generation to the next. This principle is now known as the Hardy-Weinberg equilibrium. The theory states that a population's allele and genotype frequencies are inherently stable unless some kind of evolutionary force is acting on the population. The population would carry the same alleles in the same proportions generation after generation. Individuals would, as a whole, look essentially the same, and this would be unrelated to whether the alleles were dominant or recessive. The four most important evolutionary forces which will disrupt the equilibrium are natural selection, mutation, genetic drift, and migration into or out of a population. A fifth factor, non-random mating, will also disrupt the Hardy-Weinberg equilibrium, but only by shifting genotype frequencies, not allele frequencies. Hi, if you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. All right, let's get back to reading. In non-random mating, individuals are more likely to mate with like individuals or unlike individuals rather than at random. Since non-random mating does not change allele frequencies, it does not cause evolution directly. Natural selection has been described. Mutation creates one allele out of another one and changes an allele's frequency by a small but continuous amount each generation. Each allele is generated by a low, constant mutation rate that will slowly increase the allele's frequency in a population if no other forces act on the allele. If natural selection acts against the allele, it will be removed from the population at a low rate, leading to a frequency that results from a balance between selection and mutation. This is one reason that genetic diseases remain in the human population at very low frequencies. If the allele is favored by selection, it will increase in frequency. Genetic drift causes random changes in allele frequencies when populations are small. Genetic drift can often be important in evolution, as discussed in the next section. Finally, if two populations of a species have different allele frequencies, migration of individuals between them will cause frequency changes in both populations. As it happens, there is no population in which one or more of these processes are not operating, so populations are always evolving, and the Hardy-Weinberg equilibrium will never be exactly observed. However, the Hardy-Weinberg principle gives scientists a baseline expectation for allele frequencies in a non-evolving population to which they can compare evolving populations and thereby infer what evolutionary forces might be at play. The population is evolving if the frequencies of alleles or genotypes deviate from the value expected from the Hardy-Weinberg principle. Darwin identified a special case of natural selection that he called sexual selection. Sexual selection affects an individual's ability to mate and thus produce offspring, and it leads to the evolution of dramatic traits that often appear maladaptive in terms of survival, but persist because they give their owners greater reproductive success. Sexual selection occurs in two ways: through male-to-male competition for mates and through female selection of mates. Male-to-male competition takes the form of conflicts between males, which are often ritualized, but may also pose significant threats to a male's survival. Sometimes the competition is for territory, with females more likely to mate with males with higher quality territories. Female choice occurs when females choose a male based on a particular trait, such as feather colors, the performance of a mating dance, or the building of an elaborate structure. In some cases, male-to-male competition and female choice combine in the mating process. In each of these cases, the traits selected for, such as fighting ability or feather color and length, become enhanced in the males. In general, it is thought that sexual selection can proceed to a point at which natural selection against a character's further enhancement prevents its further evolution because it negatively impacts the male's ability to survive. For example, colorful feathers or an elaborate display make the male more obvious to predators. In our next video, we'll read Section 11.2. Mechanisms of Evolution.